As Rosie said, my name is Adrian Fox. I lead the plant virology team at Fera Science in the UK. We are the national reference lab for plant health, but I'll talk a little bit about that in a moment. But we are a private government co-owned organisation run as a commercial enterprise, but we do all of the statutory testing for GB plant health, except for Scotland, because Scotland is different, but anybody that knows the Scots will appreciate that Scotland is different. So managing brown rugose fruit virus in the UK, we've had it for several years. We are still under what you guys will call eradication, but I think that as we go, we'll explain a little bit of the differences between how we approach eradication and possibly the approaches taken elsewhere in terms of when we eradicate and how we eradicate. But I suppose the subtitle of this talk is, how do you manage something like brown rugose fruit virus when it's on everything, everywhere, all at once? And I will apologise up front, there's a lot of data in this presentation. You don't need to grasp everything, just have a look at the general themes and trends, and hopefully that will help inform understanding of how we approach management and eradication of this virus. So I said I'm based at Fera Science in York, so that's in the north of England, and this is my team. We are a team of 16 now, and we do all of the import virus testing for seeds, plants for planting into England and Wales. We also do the inland surveillance testing, and we have an ongoing surveillance programme. And we have a supporting research program as well. So let's find the pointer. There we go. So a lot of our work is, as I say, dealing with import, export, trade facilitation, but also we have a suite of research projects largely around the application of high throughput sequencing in plant health and using that to better inform what we're dealing with. And also, we look at the epidemiology of viruses, but since 2018, a lot of our work has been focused on management of brown rugose fruit virus. And this is a picture I will come back to later in the talk. So just to introduce brown rugose fruit virus, as if it needs an introduction at this point, the scientific name ToBRFV fructirugosum, that's something that virologists are only recently coming to terms with, is a Latin binomial, and it still doesn't feel right. So it was first recorded in Jordan and Israel in 2014, 2015, and we had our first European outbreaks in 2018, and at the same time there were outbreaks in Mexico as well. It's problematic because it's environmentally stable, it's seed-borne, it can overcome the TM22 resistance genes that are in commercial tomato and some of the L resistance genes in pepper. And because it's environmentally stable and mechanically transmissible, it's spread rapidly through contact, so plant-to-plant contact, through tools, so general working, through clothing, via pollen on bumblebees. And it's also seed transmitted as well. So the approach we use is to look at biosecurity and hygiene measures as the primary measure to minimize spread and limit impact. However, more recently, we do have the advent of resistant varieties. That's not easy to say at this time of the morning. And I'll come back to some of those right at the end. There isn't much within this talk on resistance. But the general approach we take to biosecurity and hygiene is very similar to this diagram that was put together by Ian Mackay during COVID. And so we don't have any perfect control measure. Seed testing in its own right will not give you 100% control of brown rugose fruit virus. Using biosecurity measures on place of production won't give you 100% control. Making sure that you don't have hot fruit, so in highly infected fruit circulating, won't give you 100% control. And so what you need to do is you need to layer these different aspects of control measures up, and eventually what you will get is a high level of control by layering measures of imperfect control. And so the data I'm going to be presenting over the next 20 minutes or so is really looking at the work we did to understand how to best control the virus, so understanding how stable it is and how to to denature the virus, but also how to better detect the virus. And so I will run you through that. And then towards the end of the talk, we'll run on to some of the current work we're doing. So very much work in progress, but to give you an idea of where our thinking is going. So the current distribution of the virus, you'll now notice 2 dots in Australia from the EPO map, but basically wherever tomato is grown, you will find the virus. The one missing area is sub-Saharan Africa. There's a big question mark over whether that's just because people aren't looking, or whether the high amount of field tomato that's grown is not expressing the virus in the same way. It's not spread as rapidly to field tomato. And again, you've got some questions here with places like Peru that are absent, when we know that there have been seed detections from Peru, but no plant detections reported as yet. The current situation in England and Wales: brown rugose fruit virus is a regulated quarantine pest. So from the Australian response, that would be, we are still in eradication. And under UK legislation, we can take action on suspicion of the presence of regulated pest. So what that means is, when we get a seed positive, if we are confirming that seed positive using multiple tests, if we interpret our tests as being positive 3 times in a row, then that's adequate suspicion to take eradication measures. We don't do the bioassay that the Seed Federation would request. But also it means that our inspectors can go on sites to do inspections and then see if they see symptoms, or we can take an annual survey, which we do. If we find the presence of the virus, that crop is allowed to run for the year. The grower can continue to market their fruit if it's marketable, and then their requirement is to eradicate the virus before the next growing season. Our first outbreak was in mid-2019. We've had 2 to 3 outbreaks per year since 2019. However, we've also got 2 to 3 outbreaks per year being eradicated and coming off that list. And we know that because we do annual surveillance of all of our tomato glasshouses. Nationally, we have, I think, 60 premises and around 80 crops that are grown. And the big advantage we have is that we're not Almeria, we're not the Netherlands. We have a very dispersed industry. So we also have growers who have been through potato spindle tuber viroid, calumnia latent viroid, pepino mosaic virus, clavibacter. And so what we also have is a group of growers who are incredibly savvy when it comes to biosecurity measures, and have just continued to rack up their biosecurity measures as we go. So, for example, we have one grower where their pack house for their 6 growing operations is about 40 miles away from their nearest glass house, so that you've got no circulation of fruit from any of those sites back into the glass houses. We also have an outbreak on the states of Jersey and the Channel Islands, but because they're not officially under the England and Wales NPPO, that's classed as separate to UK. And there's been a recent outbreak in the Republic of Ireland as well. So early on in brown rugose fruit virus, we wanted to look at the survival of the virus so that we could better understand how to eradicate it. And this work was funded by growers. So what we wanted to do was give information back to growers that was useful and usable within their biosecurity protocols. So one of the first things we did was to look at survival on skin and gloves. And this is quite a simple experiment. You infect somebody's hands. So either you rub them in contaminated sap, or you just rub the hands against a leaf. And we did this for skin and gloves. You then swab that, you swab it onto a plant. These are test plants of Nicotiana tobacco. And then you see if you get the development of these local lesions, and that indicates that you have viable virus. And basically at 2 hours, you still had contamination on the hands that could spread the virus into a non-infected plant. And there was no difference in those results between sap contamination or leaf contact, so just brushing against a leaf. You still had over 2 hours. I say over 2 hours because we stopped the experiment at 2 hours, because we thought it was inhumane to keep people without being able to use their hands for the rest of the day. So we stopped that experiment there. But I would have thought that you would continue for several hours beyond here, where that virus would remain stable and active. And so from this, what we advise is that rather than, we do still advise hand washing on entry to the glasshouse, and that's for control of a whole host of other contact transmitted pathogens. But when it comes to brown rugose fruit virus, we advise using gloves and changing them often. We looked at the efficacy of hand washing, and what we saw was that a quick 30-second hand wash with a medicated hand washing solution, so that's the pink Hibby scrub that is used in surgery, after 30 seconds you still get positive for infectious virus on the hands. We looked at different treatments, and effectively what happens at 60 seconds is you get a reduction in infectivity, but you don't remove the virus completely. And so what I would say is this again shows that hand washing is ineffective. My favorite thing that I've worked out so far, so for COVID, if you want to measure 20 seconds of hand washing, you sing happy birthday. If you want to measure 60 seconds of hand washing, if you start singing Queen's Bohemian Rhapsody, by the time you get to the point where Freddie Mercury says, "Mama, I just killed a man," that's 60 seconds. And so what I do after this webinar is challenge you all to go press play on Bohemian Rhapsody, wash your hands for 60 seconds and see just how long that actually is. This is unworkable for entry to a glass house. So again, wear gloves, change them off. We looked at heat inactivation of the virus, and this was with a view to informing growers on how to disinfect picking crates. And so 90 degrees, 5-minute soak and some Virkon, and you should be free from the virus. When we did the heat inactivation experiment, we again found 90 degrees. So if you're heating things like picking trays to more than 90 degrees, then you will eradicate the virus for those trays to move back into glass houses. And we have one grower that's effectively built a large autoclave on his site for his picking crates, where he's used a shipping container with steam pipes running into it, and that's how he maintains virus freedom on his sites. We also looked at survival on glasshouse surfaces. So this was with a view to looking at, if we eradicated a crop, how long would that glasshouse have to remain fallow or with an alternate host in there before you could actually move to say that you had eradicated. And up to 7 days, everything is positive. Up to 3 months, most things are positive. And at 6 months, you're still getting infectious virus coming off several surfaces. And so again, a fallow period is probably of no use. Swapping to alternate crops, so moving from, say, tomato to cucumber, would give you a growing season where you were free of the virus. However, I also appreciate that moving to a cucurbit brings its own problems with things like the CGMMV. And the other thing to remember, we have looked at effective disinfection, and things like Virkon will work, things like glutaraldehyde solutions will work. However, even when you have exposure, so Unifect G for 10 minutes was effective, you can still detect the virus using molecular methods. So here what we did was we disinfected the surface with Unifect G. The no treatment control is just the no disinfection, and then that's swabbed onto a test plant and you get the passage of the virus. And here with the disinfection, you're detecting the virus at around the same levels that you would get for the no treatment control. However, post disinfection, it's not viable virus, but it is still detectable. And this is one of the challenges we have with molecular diagnostics, and I'll come back to this later. So in terms of the diagnostics we use, we follow the EPO standard method, so PM7146. And what this includes is recommended tests, so 2 conventional PCRs and 4 real-time PCRs. However, it does include what we call other tests. And so these are not officially recommended for use, but they are to raise awareness that these tests are available. And so you have things like serology. We have the bioassay, which is currently our only way of demonstrating viability. However, there are challenges with that in terms of routine use. and high throughput sequencing. And certainly some laboratories such as the NIVIP, the regulatory laboratory in the Netherlands, sequence all of their brown rugose fruit virus findings. But there are some diagnostic challenges. There are interpretation differences between laboratories. So some of this is to do with thresholds and cutoffs. So we know that one of the large labs in the Netherlands, for instance, uses a cutoff. So anything over, say, a 32 CT value on real-time PCR would be considered to be negative. We don't use a cutoff. What we use is multiple tests to give us further information on finding. And as I said, PCR-based detection picks up both viable and non-viable virus. And this is where I circle back to what I said about UK legislation, that we can take action on suspicion of the presence of the virus. So because it's a quarantine pathogen, we don't actually care particularly whether that detection is viable or not. It shows that the presence of the virus is there. But we also found that there were some strong positives with no symptoms at the time of sampling. So what I'd say is that plant symptoms are not a reliable measure of infection. You can get perfectly healthy looking fruit that tests as a really strong positive of a plant. But we also found weak positives. So these are what we call high CT values. And that's across multiple tests that could not be confirmed through a secondary method. And so the question there was, is there a biological issue that's working against us? Have we got environmental residues or laboratory contamination? And I'm very careful to use the words environmental residues, because if you just talk about contamination, laboratories can get quite defensive, because that suggests that the contamination may be coming from within the laboratory. However, with this virus, as I'll present, there is a huge problem with a buildup of residues of the virus in the wider environment, and especially in somewhere enclosed like a glasshouse. And so labs like ours take extreme precautions against contamination. I will talk later, if anyone is interested, about the levels of biosecurity control we take when we're dealing with a single sample. But so I don't ruin the flow, I won't go into that just now. So Anna, in my lab, Anna Skelton, initially looked at how we optimize plant sampling. So as I said here, is there a biological issue working against us? So the first question is, are we picking the right bit of plant? So this was collaborative work between ourselves and the Dutch, so the Dutch Regulatory Lab and Wageningen University. And it's based on a classical piece of plant virology from the 1930s. And so Samuel, back in 1930, did this piece of work where he put TMV into a tomato and he watched it spread to the roots first, up to the top of the plants, and then go fully systemic within, say, 18 to 25 days. When Samuel did that, real-time PCR, modern diagnostics hadn't been invented. And so he did this by cutting the plants up into 2-inch segments and rubbing each of those segments on a tobacco plant for that local lesion assay. What we wanted to do was to see whether you got a similar pattern of distribution of the virus. And so could we go from there to giving inspectors advice on what to sample? So we did inoculation experiments where we looked at both young plants, so about 4 weeks post-emergence. So that's looking at plants within the propagation house. We looked at plants when they enter the production facility, so about 8 weeks old. And then we looked at plants once you'd got truss formation and fruit set to see whether that pattern of emergence throughout the plant changed as the plant got older. And we also looked at outbreak sampling. So the Dutch went into active outbreaks to look at the presence of virus there. And so this schematic just shows simply the first DPI, so the first days post-inoculation, when we found the virus in each of these places. And so what you'll notice for the young plant, this follows that schematic that Samuel did back in 1934, that you get the plant, the virus at the top of the plant, then within a week it spreads back to the middle, and then by about 3 weeks, it's gone into the mature leaves at the bottom. And what this does, it follows this hypothesis we have in virology called the source sink hypothesis, that the virus is moving from wherever the source is, so the inoculation point, to the sink, which is wherever the plant is actively putting its efforts into, whether it's foliar growth or fruit formation. So as I say, here you go from the inoculation point to the active growing tips, whereas once you've got fruit formation, you go from the inoculation point, and you'll notice it goes quickest to the nearest fruit. And so from that, what we did was we gave advice to inspectors on when you go into a crop, you don't just take a standard sample. What we want is the inspector to actually think about the sample they're taking. So before you get truss formation, you want to be going for the actively growing tips, preferably the top of the plant, but you may have side shoots as well. And as the plant gets older, we ask for samples of sepals from the plant because that gives us the best chance of detection. And this was validated by the Dutch, who did their outbreak sampling and found the same results that we found from our controlled experiments. As with you guys in Australia, we worry about our seed interceptions. What I would say is that the number of seed interceptions we get of brown rugose fruit virus, although I still believe that seed transmission is a risk, I would like to praise the seed industry for the efforts they've made in actually getting on top of this virus and helping to clean up the system. And as you see here, so back in 2022, so this is out of about 350 seed lots per year. So it's still not a huge amount of interceptions if you're looking at 9 or 10 positives for that virus out of 350 tomato seed lots. But you'll see that we had a few pepper seed and some tomato seed. Through 2023, that's dropped. And last year, we only had a single interception on tomato seed of the virus. So as I say, I still believe that seed does pose a risk, but what we need to do is look at other sources of the virus as well. We'll take a sidestep just to think about how we determine a positive and a negative. It's important to remember that when a laboratory tells you that something is positive or negative, that's an interpretation of data. So for example, so even though we have EU emergency measures and an EPO standard, not all laboratories will report the same sample as positive, because you can harmonize even the test method, but because of the way that the results are interpreted, that could go one way or another within the laboratories. So you get differences in test result between labs, especially on seeds. Some of that is technical differences, so the way the samples are prepared, human error, RNA extraction methods, PCR kits, machines. So what my machine reads and what the machines at Knacktime Bow or AgriBio read may be slightly different. You then get slight differences of interpretation in C2 and CT and CQ values. I'm not going to go into this. This is a proper geeks holiday bit. But so basically where we set the baseline to interpret what's positive or negative may be different depending on which lab you're in. Some laboratories use a cutoff. As I said, we don't. There may be contamination. And what we're still trying to work out is the response of plants that have intermediate resistance. So this is the suppressive response of the plant. So does that look the same when a seed lot comes through or when a plant comes into the laboratory as one of our positive plants? So what you've got to do is look at what your validation tells you. And so for us in the UK, a positive is target amplification. So that's positive across 3 different tests. And I'll talk about which tests we use later. And then you've got to look at the context of that test. So I would expect a leaf sample to test with a different level of positive to a seed sample, to a water sample, to a swab sample, to a fruit sample. And so to give you an idea, the number of cycles is how many PCR reaction cycles you run in your machine to indicate whether you have a positive. And on TACMAN, the positive is denoted by the rise of this line as you get fluorescence in the reaction. We would expect a seed sample to be coming up somewhere here in the mid-20s and low 30s. Our swab samples usually come up in the mid-20s. Our hottest fruit sample was about a 3. The machine actually struggles to work at that kind of level of positive. So that gives you an idea of the difference in dynamic range that we're dealing with. And just to give you an idea, each 3 CTs is about the equivalent of a 10-fold serial dilution in virus concentration. It's not exact, but that's an approximate idea to give you an idea of the dynamic range of the virus interceptions we have. You've also got to look at how reliable your sample is. So our sampling regime is based on ISPM 31, as is the Australian sampling regime. And so here is the results from Australian sampling on the right-hand side. And so we set our testing at 3,000 seeds. So the reason for that is if you look at the 200,000 plus in the international standard for phytosanitary measures, if you look for a 95% detection at 0.1% confidence, you get 2,972. Well, it's much easier to tell an inspector to take 3,000 seeds than it is 2,972. The 20,000 seed level is to get way beyond 99% confidence of probably about a 0.2% level of detection. And so if you look from this piece of work done by Fiona Constable's lab in Victoria, if you test at the 3,000 seed level, we accept that we are missing some positives of both tabamovirus and posbiviroid. Testing at the 20,000 seed level gives you a much better confidence that you are picking up infection if it's there and you're not missing these infections in this grey area. What I would say is our testing is based on pragmatism, because the laboratory can only deal with 3,000 seeds of each seed lot coming in, because we test all solanaceous seeds that are entering the UK, even if they've been tested offshore. The other thing we've got to consider is brown rugose fruit virus in imported fruit. So on the left-hand side is data from the UK, and on the right-hand side is data from the US. If we look at the UK data, 60% of the fruit that is on the supermarket shelves in the UK is imported, and it's generally imported from Morocco, Spain, Italy, and the Netherlands. And when we looked at this in spring 2023, when you've got nothing but imported fruit on the shelves, what you had is some nice looking red tomatoes, some that had a little bit of discoloration, but most of them looked perfectly fine on the shelf. Yet a high proportion of them were positive for virus. And what we've said is that if we got a strong reaction on our testing, we assumed that was from an infected plant. And so that's this red portion here from Spain, Morocco, and Italy. If we got a weaker detection, then that could be from an infected plant as virus is building up, or it could be just contamination in the processing system because there's so much virus about. But overall, around 80% of the fruit we tested was positive for the virus. I have to say the Netherlands was underrepresented in this testing, and I've deliberately used the blue and the yellow as our colouring because the Netherlands was underrepresented because of the war in Ukraine and the effect that had on energy prices, which reduced the amount of winter planting in the Netherlands that year. So I think there's an underrepresentation of the virus levels there. But we've linked into the Dutch to work on source tracking some of our outbreaks in the UK. And so we have our first outbreak, and this is all done on whole genome and put through this computer program called NextStrain. We've had a recurrent outbreak that's now eradicated. I'll talk a little bit about that in a second. And so we could trace that that was the same outbreak because the genotype of the virus was identical and quite unique. There's also this cross-protection genotype that was illegally spread in the Netherlands, and we've got these seed detections from seed from Peru as well. So what it allows us to do is to do traceback and try and work out the origins of the virus. But what it also means is that we can do outbreak source tracking and see where our outbreaks originated from. And we do feel that some of our outbreaks are related to the presence of the high amount of infected fruit within our market system. So a fruit would be considered a low risk pathway, but when 60% of the fruit on your supermarket shelves is infected with the virus, I'm not sure that that same low risk can be considered to apply. And so we've got these three cases here where we've got one that's related to this cross-protection genotype. That's not yet been detected from seed. It's a fruit origin potentially, and we don't think the grower intentionally spread that virus because he's our largest grower, has about a third of the UK-based production, and we haven't found that in any of his other sites. So we think that that is originating from fruit. Our recurrent infection, so this is in the Midlands. The grower initially identified his packhouse staff moving to the glasshouse during COVID as the source of the infection, because his first infection turned up about 4 weeks after that. But because that genotype is across several European countries, it's much more difficult to track down. And then this case in 2023, where we've had a genotype that we've only found in fruit from Morocco. And so again, there's a question there of whether that's originating from fruit as well. So as I said earlier, I think the risk from seed is still relevant, and from the UK perspective, we should consider imported fruit as a potential risk as well. And that brings us to, how do we protect our sites when the virus is on everything, everywhere, all at once? And so what you have to do is consider the relative risks within the production scheme. You've got your seed imports, you've got your propagation house, you've got your transport system, you've got all of the activities when you're working, whether that's pollination, cutting, watering. You have your staff who may be moving from pack house to glass house. You've got things like your picking crates that will move around. You've got the staff canteen, you've got the managers. And one of the things we find is managers tend to be the worst people for moving between sites. And we have the imported fruit, so you've got to consider the fruit that the staff in the canteen are eating. Could you provide fruit for those staff so they're not bringing fruit from off-site onto site? And the one that nobody talks about is that this virus is so robust, it will follow through into the waste system as well. So to give a bit of background, as I said, the UK isn't like some other areas. So this is our outbreak site in the West Midlands. He was infected for 3 years as a recurrent infection and then managed to eradicate in year 4. But what you'll notice is that he's got 2 8-hectare glasshouse blocks, and this is a solar farm that's next to it. And you'll notice there's no other tomato production around. If you contrast that to somewhere like Almeria in southern Spain, where you have plastic tunnel after plastic tunnel, and each of these plastic tunnels is about the same size as one of those glasshouses on the Serra. So what we don't have is that risk of the virus going from site to site directly, and that's something we use to our advantage. As I said earlier, we have the issue with the virus being detectable even when it's inactive. And so what we need to think about is, when we get a detection, what's the biological relevance of that? So from the seed industry perspective, is it viable or non-viable virus? Are you just picking up the traces of virus that have been inactivated through the seed cleanup system? But from my perspective, within the glasshouse outbreak, you've also got to think about, is that virus that you're picking up environmental contamination? And I'll just show a little bit of data as to what I mean by that. So this is our active outbreak site that was active for 3 years and now eradicated at the Serra. And here you can see the plant death. And as you go further into the crop, it actually looks relatively healthy. And the grower managed to get crop off it for 3 years. He estimated that he lost about 30% yield year on year. But when you went into the glasshouse and you used a cotton swab to swab areas where plants hadn't touched, you could still get positive PCR tests from those swabs. So here you've got the beehive that's used for pollination coming up with a 17 CT. We'd expect a positive plant to be around that. Again, reaching down inside the fan units and under the gutters, you're also getting really strong results. At this site, we think Glasshouse 2 was the first one infected and Glasshouse 1 was after, which explains why you get stronger results from Glasshouse 2 to Glasshouse 1. And my favourite one here is this one that's named Tyvek Suit. And that was Anna from my lab who went up on the scissor lift to swab the very roof of the glasshouse. And when she swabbed that, we noticed there was a rusty mark on the back of the suit from where she'd been leaning on the scissor lift. So we swabbed the rusty mark. And from that, we got a CT value of 16. So again, somewhere that we'd expect an infected plant to be. And then you start considering the wider literature on brown rugose fruit virus and its relatives, such as TMV and TOMV. And you get papers like this from the 1990s, where they found infectious tomato mosaic virus in fog and clouds. And this is where we then start talking about environmental residues. There's dust that are produced by plants, whether it's trichomes, whether it's pollen, Evaporation from water may play a role. And so what we want to do is try and understand how all of that environmental contamination can lead to further infections. So work we did with Natasha Mekle in Slovenia, looking at water contamination. So you have an infected plant, you put some healthy plants into rock wool in the water, and can you pass on the virus? So what you see is after about a week, you're starting to get a virus released into the water, and those virus particles are released from the roots and they remain infectious. And there's further work that was done to show that they can then contaminate plants within the irrigation system. For organic growers and those working in soil-based systems, Mary in my group is looking at soil survival. And so we grow plants in soil, infect them. We keep the soil with root residues and all. Some of it we sieve, some of it we and plant on. We then grow further plants and we also bioassay from that as well. And we directly test it. And what we're finding is that the direct PCRs from the soil and the extracted roots are positive, certainly up to 70 days. We're saying not applicable here because we couldn't actually get any root extract from it because they'd rotted within the soil, similar with the bioassay. And our bait test is currently negative. What I would say is we've done a full-length genome amplification from this. And so even though the virus is at 18 in the soil, we think that you have a full-length genome, potentially viable virus, but it's below the levels at which our bait test can pick it up. And Amy in my group is then looking at how the virus goes airborne. So how is the virus released from plants? And so can it be evaporated? So we use this highly technical system of a clip seal sandwich box. Here we've got detached leaves in the bottom, but we've also used just a straight lid off a laboratory tube and evaporated virus from that. And what you find is that you can get positive virus on the top of the lid after a long weekend. And in the plant growth chambers that we built, you can get virus residues on the roof and the sides at levels that would certainly be considered contamination. And if that was a seed lot, the 27 would be considered positive. And so I think we've got evidence for release from plants as well. And so what we're doing now is looking to confirm that, and then we'll get that published because I think that's an interesting result as well. So finally, just to say the molecular test we use at Fera, as I say, we don't use CT cutoffs. What we do is we use multiple tests. And these are all the tests that are mentioned within the EPO standard. And the ones we actually use at Fera are these. So we have the 3 real-time PCRs, which cover the different parts of the genome. And so we can say for definite that we have different sections. And we also, where possible, use the conventional PCR for confirmation as well. So we actually have 4 different parts of the genome, so we have good genome coverage. And then, and only then, will we report a positive. And the final piece is that Lynn, a PhD student in my group, is now looking at tiled amplicon sequencing as a method of determining that whole genome amplification. And this is work that we've been doing in collaboration with the guys at AgriBio. So Dave Lovelock, when he was based with Fiona Constable there, gave us some brown rugose fruit virus primers. Lynn's designed her own as well. And what we're doing is basically looking to see if we can equate an intact genome with viable virus. But hopefully I can come back at some point in the future and talk more about it. As I said, the important thing now is considering what we know about the survivability of this virus and the environmental residues and the ability for it to carry over between outbreaks, is to consider those relative risks to keep the virus out of your production if possible, and if you've had an outbreak, to eradicate effectively, which is possible, and then to make sure the virus doesn't get back in again. So your take-home messages: brown rugose fruit virus is an environmentally stable virus. It's resistant to heat and chemical disinfection, but eradication is possible. Play to your strengths. As a nation, you have an intrinsic understanding of high biosecurity measures at both a national level and a company level. Use that to your advantage. Resistant varieties do offer an exit ramp, but they're not a magic bullet. Understand what those different resistance mechanisms are, whether it's high resistance or hypersensitive resistance or intermediate resistance, effectively some kind of suppression. But the important thing is to protect resistance with good biosecurity. It's not a magic bullet. Thank you for listening.